This video is about the individual assignment on computer-aided drug design subject, ISBM 4021. Time using BPsp 82. Basic Local Alignment Search Tool is an algorithm for comparing primary biological sequence information, such as the amino acid sequences of different proteins or the nucleotides of DNA sequences. The BLAST search enables the researcher to compare a query sequence with a library or database of sequences, and identify library sequences that resemble the query sequence above a certain threshold. Different types of BLASTs are available according to the query sequences. For example, following the discovery of a previously unknown gene in the mouse, a scientist will typically perform the BLAST search of the human genome to see if humans carry a similar gene. BLAST will identify sequences in the human genome that resemble the mouse gene based on similarity of sequence. First, open the BLAST website, http://blast.ncbi.nlm.nih.gov/blast.cgi. Paste the FASTA protein sequence on the box provided in Protein BLAST. Make sure the algorithm is side BLAST. Then, click BLAST at below. Then you will get conserved domain, graph and alignment. This is the conserved domain. This is the graph. And this is the alignment. Interpro is a resource that provides functional analysis of protein sequences by classifying them into families and predicting the presence of domains and important sites. To classify proteins in this way, Interpro uses predictive models, known as signatures, provided by several different databases referred to as member databases that make up the Interpro consortium. InterproScan is the software package that allows sequences, protein and nucleate to be scanned against Interpro's signatures. Signatures are predictive models, provided by several different databases referred to as member databases that make up the Interpro consortium. InterproScan is a tool that combines different protein signature recognition methods native to the Interpro member databases into one resource with lookup of corresponding Interpro and Go annotation. To use InterproScan, open InterproScan website. Then, paste the FASTA protein sequence in dialog box and click Submit. Functional domain will appear. This is the domain searched using InterproScan. ClustalX is a Windows interface for the ClustalW multiple sequence alignment program. It provides an integrated environment for performing multiple sequence and profile alignments and analyzing the results. The sequence alignment is displayed in a window on the screen. The versatile coloring scheme has been incorporated, allowing you to highlight conserved features in the alignment. Using this software, you can cut and paste sequences to change the order of the alignment. You can select the subset of sequences to be aligned. You can select the sub-range of the alignment to be realigned and inserted back into the original alignment. Alignment quality analysis can be performed and low-scoring segments for exceptional residues can be highlighted. This is the .dnd format that opened with Mega Software. The Protein Data Bank (PDB) archive is the single worldwide repository of information about the 3D structures of large biological molecules, including proteins and nucleic acids. The PDB was established in 1971 at Brookhaven National Laboratory under the leadership of Walter Hamilton and originally contained 7 structures. After Hamilton's untimely death, Tom Coetzel began to lead the PDB in 1973, and then Joel Sussman in 1994. In 1998, the Research Collaboratory for Structural Bioinformatics (RCSB) became responsible for the management of the PDB. In 2003, The WWPDB was formed to maintain a single PDB archive of macromolecular structural data that is freely and publicly available to the global community. It consists of organizations that act as deposition, data processing and distribution centers for PDB data. This is the primary structure of MyProtein. This is the secondary structure of MyProtein. This is the tertiary structure of MyProtein.